Hello, everybody. This is Joel Rojas, and I am part of the policy team. And today we're going to be talking about a very important and also um, hello, everybody. This is Joel Rojas here. I am from the Dominican Republic chapter, and I am also part now of the policy team here at Given for the next COP that is going to be celebrated in Cali, Colombia. And today we're going to be speaking about a very important topic that is going to be discussed and negotiated in the next COP, and it's no other than, sorry, little moment here. Technology. So we're gonna talking about biotechnology. We're gonna be talking about agenda item number 5, which is on regards of synthetic biology, its use, and also the normal and current concerns, situations, and also voices are arising because of their use and the rights of the people that are involved in this topic. This is a topic that is not new and emergent. It has been in the table from some time, but now has taken more and more power because of the implications of their use in developing states, but also in traditional land. And the new technologies that are arising are placing as well some challenges in the definitions that we have right now for other organisms that we know in the Convention of Biological Diversity, CBD, which are living modified organisms. So today we're going to be speaking a little bit of synthetic biology and why this topic is important in the context of the CBD and in the context of Given. So, first of all, let's define what is synthetic biology for those who might or might not know. Maybe you don't have a background in biology, a background in molecular biology and genetics. So let's talk a little bit about what is SynBio, which is going to be the name that we're going to put to synthetic biology into this little webinar today about synthetic biology. And as it has been described in 2015, it was described as a further development and a new dimension of modern biotechnology that combines different areas, which are science, technology, and engineering. This has been done in order to facilitate and accelerate the understanding, design, redesign, manufacture, and/or modification of genetic materials such as DNA, RNA, proteins, etc., and living organisms, but all of this also probably biological systems. This is the definition that we have at the moment. And of course, this relies, Symbio, on a suite of supporting technologies and tools, and some of which are also used to establish some protocols and also processes in the field of genetic engineering. So, as you can see, it's a very broad definition, but is this actually the definition that we have for synthetic biology? The answer is nobody knows how to define synthetic biology. In fact, actually, in the recommendation that was placed in SBSTA for the potential multidisciplinary expert group in synthetic biology, one of the considerations that they are putting in one of the terms of reference point items is to consider important and outstanding issues, meaning issues that have been in the table from a long time, which is an operational definition of synthetic biology, so we can relate this definition to the Convention of Biological Diversity and to the objectives of the Convention of Biological Diversity. And we actually might know what is synthetic biology at this point. It's just that as of right now, technologies are evolving so fast, and the products of synthetic biologies are being developed and released at such a rate that for us it's kind of difficult to make a current definition that is going to serve for many, many years or for a brief period of time of that served for previous applications that were placed into the market before. And with this, we can move to maybe to see what everyone thought was synthetic biology in one period of time. And actually, sometimes this is synthetic biology up to some extent. So what happened is that in some occasions, in the use of synthetic biology, meaning genetic engineering, we take the genomic DNA of a given cell, of a given tissue, organ, maybe from a species, maybe from a group of species, a complex species. We sequence this, we put this in databases, and with this, we're going to talk about another issue, which is the SI. This is for another webinar. And with this information in the databases, we can analyze basically the genome or genes of different species, look at what will be the interactions and effects that we can have by modifying these inside of the same group of organisms, individuals, species, etc. And with this, we can do different things. We can create new genomes. We can also create new sequences, or maybe modify existing sequences in organisms without the need of transferring genes from other species. And we can basically create, although we don't like the word create, we say synthesize new cells, new organisms that have different configurations than the wild species or wild types that we can find in nature. But this is not the whole story. I mean, synthetic biology has been seen more extensively in different applications. And again, sometimes it could also be this, then as I found in this, for example, paper of Yen of 2023, actually, the applications are endless. I mean, and this is defined with medical and pharmaceutical fields, but also some of these applications are now starting to be used in the biodiversity field, and this is where we have to start to pay attention about and understand which might these technologies. I mean, we have, for example, expression fine tuning, basically enhancing the expression of genes that we want to enhance, to upregulate in order to have different products in the same species. We can have, for example, machine learning, which is one of the concerns that is arising more and more because of the heavy data input that you have to put into place and the lack of transparency and also governance of this data. Where is this data coming from? How did I obtain this data? Did I ask for consent for this data? For example, a local community who is a custody of biodiversity, did I ask for permission for different stakeholders, for the authorization for governments for the use of this data in order to create these probably new synthetic genomes or modifying existing genomes? Did I ask permission for this? Also, protein engineering, how we can improve proteins, because as you might or might not know, proteins are basically what moves organisms. It's like the powerhouses of different processes, and without proteins, basically, we're doomed. We are made of proteins, and proteins do a lot of work. So protein engineering, meaning creating new proteins in order to see if they have different appliances, also is something that is done with synthetic biology. And also something very important is CRISPR-Cas systems, because this will help us and are allowed to help us to understand also what might be of difference between GMO, genetic modified organisms, and LMO, a living modified organism, and a symbio product, which might or might not differ from the current knowledge that we have. And with this, we have to pay attention to two different concepts, which are still debated until this day, which are GMOs and edited organisms, or organisms that come from CRISPR, for example. What happens is that with GMOs, usually, or most of the time, or maybe all of the time, you may have a foreign gene that comes from another species, and this will be transferred and transplanted to another organism. This why it's called a transgene, because it comes from another species and is implanted into our species. With this, what we want to do is to express this gene, meaning this protein or this product, into this species. And that's what we call GMOs usually. And this has been since Cartagena, I will say since Asilomar conference, something of concern, something of to discuss because of the precautionary approach. We don't know how the species might react in nature. Now we know which might or might not be the safest standards. Different countries have different regulations. Some states allow GMOs to be into their market and soil. Some countries no. It depends. In CRISPR, however, the story is very different because with CRISPR, we have a cut inside of the gene. And usually, what this does is to maybe silence the gene and remove it from working, basically. By silencing the gene, you are preventing something to be expressed, meaning that you're preventing something to be inside of the cell, inside of the tissue, inside of the organ that might be a target for maybe crop improvement, pest prevention, etc., etc. With this, when you modify this with CRISPR, the main issue is that the cut is so exact that sometimes might look like something that comes from a natural population because species all the time and populations all the time change their allele frequencies. You can have mutations, for example, that come randomly inside of the population, and these mutations can look something like the result of a CRISPR gene editing process, meaning that, for example, you can have a precise cut and you can have there will be uh, a deletion. You can also may have an insertion, maybe a nucleotide got inserted into this DNA. And when you make the cut by the repair mechanisms that the DNA have after the cut. So with this, we actually cannot tell when an organism comes from a modification from CRISPR or not. And this is a problematic because we cannot, if we cannot tell which came from natural or from CRISPR, we cannot make safeguards, we cannot make approaches to assess and to manage, for example, the risk that might come from these modifications. And basically here in this slide, I'm presenting how more or less works. This is basically a little overview of how the CRISPR-Cas9 system works. Actually, the CRISPR-Cas9 system was not synthetically made. It actually exists already in bacteria from millennia. And basically, it's a defense mechanism against the viruses of the bacteria, which are phages. What we do, for example, with CRISPR is that we unite two different things. We have a guide RNA, which is gonna be like our like the one that says where do we need to make the cut, and then we're gonna have a protein which is Cas9, which is the protein that makes the cut. Look Cas9 like the scissors, which are gonna make the cut into your gene. So what we're gonna do in this process is that here we are gonna create a RNA, which is a single guide RNA, which is gonna guide, as its name says, our protein Cas9, which is gonna be the scissors, into my gene. The guide RNA is gonna detect a region which is specific, which is called the PAM sequence. It's very short. And with after detection of this sequence, the RNA is gonna bind nucleotide by nucleotide, meaning base by base, into the gene. After it matches, then Cas9 is gonna create a cut, and this gene is gonna be take out of the DNA. Here, we have different methods of repairing the DNA because, of course, if you cut DNA, then it's going to be open. DNA is not very kind of going or inclined to be cut and left out because that will make problems later on. And what we have is the repair. This way, for example, non-homologous end joining is basically like you put the two pieces together. We may have homology directed repair, which with this we can insert some genes, which will also be like a transgene in that context. But these are other ways. In this way, we just like put the two pieces together again of the DNA, and that's it. With this we can have deletion of one base, insertion of one base, and with this we can have different things. But what we did here is basically cut our gene and basically silence it, so we don't have the effect of this gene. And these could serve as a base to understand gene drives, because one of the main issues right now in the CVD from different stakeholders, not only parties, but also different organizations, IPLCs, women and youth, is the use of gene drives in the wild. So what happened is that gene drives are patterns of inheritance that go beyond Mendelian laws. Basically, in the normal Mendelian frequency of transmission of characteristics of genes, we have here the normal inheritance is 50-50 for an allele. Like we receive 50 from our mother, 50 from our father. In nature, alleles work in a stable population under the Hardy-Weinberg equilibrium. We have that each allele might follow 50 and 50 if we don't have different things going inside. So this is a normal inheritance. The unaltered gene might not spread that easy into a population. What happens is that with gene drives, Mendelian laws are loose, basically. And with this, we have more than 50% of transmission of a desired allele. So basically, we can have almost 100% of transmission or a secure transmission of an allele. And how this works is very easy. So what we want to have is here, in the case we want to use gene drives, of course, a gene drive cassette, which is going to be composed about our guide RNA, the one that we explained before, Cas9, and a cargo that can mean different things. This will, of course, cut the guide RNA, and the Cas9 are gonna cut any gene of interest. So what you do usually is that you make a gene drive parent, let's say a mosquito, which is one of the applications that we are seeing right now. You make a mosquito modified with two alleles, meaning one from the one from the father, that have here this cassette. And we're gonna cross this with a wild type parent, meaning a mosquito from the nature. When we have this, what we're gonna have is that actually the mosquito, the son, the daughter, is gonna receive one from the father and one from the mother. So after this gene drive cassette, as you can see here, actually is gonna cut the other allele that received from the other parent, and it's gonna insert this cassette in that allele, meaning that it will be expressed in this organism. And through gene conversion, we're gonna have this effect. So we're gonna have the cassette that came from one parent, and we're gonna have the other cassette that is gonna be coming from this first one that we have by a cut and conversion through homology directed repair, which is the other mechanism that we explained before. And this is an issue because with this, we have an ensured transmission of the gene cassette that you want to transmit into the next and the next and the next and the next generations. And as we know, for example, some species have more fast pace of transmission of inheritance, like for example, insects. So this could be easily transmitted through entire populations of mosquitoes, of flies, of different organisms, of bees that we do not have a way to control in some occasions. So what is a SynBio product actually? Because we saw that actually we have an issue with the definition of SynBio. So defining what is a SynBio product is way more difficult. So in 2017, the SynBio ACTEC actually have some discussions about this. What will be a SynBio product? And actually, they concluded that at that time, in 2017, most living organisms already developed or are currently under research. They fail under the definition of LMOs under Cartagena Protocol. And those organisms included organisms containing engineered gene drives. The problem is that this has been now a little bit challenged. So we still use this deliberation, and we have seen that after this, in different symbiote groups, we have the same conclusion. But it's now starting to be a little bit challenging because of the technologies and because of the applications that we're starting to see of these products inside of nature. So they saw that actually techniques that involve cell-free systems did not result of living modified organisms, meaning that these are not regarded as much as LMOs, but also there were different interpretations and different interpretations regarding epigenetic modification and also other uses of other technologies might or might not be regarded as LMOs. So this is starting to be a problem for some parties and for some stakeholders and organizations and groups, of course, and right holders. And in the years, we have different technical expert groups discussing different things. The first one was in 2015, the second one was in 2017, and the next one was in 2019. So the first one in 2015 actually discussed what is in bio, what are the potential benefits and risk, and the impacts, the potential impacts that we can have of these organisms. After this, we have another designation by the COP to another ACTEC, which said which are the recent technologies that are used at that time. And we saw also which might be these organisms that fell or not under the definition of symbiote product. And also we have recent organisms development and more benefits, risks, and impacts. And in 2019, we have the first review of the current state of knowledge, the potential positive and negative impacts in the environment, and if new organisms are to be considered outside Cartagena protocol. And this was not enough for a lot of reasons. Now, right now, we don't have the operational definition of symbiote still. We still are analyzing the potential benefits and risk, most of them the risk that we can have, assess the impacts the is going to have in biodiversity. And this actually is very important to be continued because technologies evolve with time and new organisms arrive. So this is a constant work, and in fact a new kind of technical expert group arise through decision 1531. And this new group was called multidisciplinary because it involved people that work with synthetic biology and have background in biology and other areas, but also included people who might not have expertise in specifically in the field, in the lab, let's say, but they have the knowledge of the implications of what these organisms have. And in this group was given a duty, and the duty was to establish a process basically for broad and regular horizon scanning, monitoring, and assessment, meaning understanding which is the recent, the a little bit of the previous, but the recent in the short, medium, and long term applications, technologies, and so on about synthetic biology, monitoring this, and how we can assess this new developments and trends in synthetic biology. As you can see, there is a guy here representing the youth. Uh, and in this group, we uh, develop different outcomes. One of them was that we identify, for example, in the group, I'm going to talk as we because I was there, and we identified different trends and issues. From there, we took five that were selected in an expert-driven way. So we took them for a deeper assessment. So these five priority items were selected because of potential recent and future concerns, issues, and risk that might arise from from them. So these five were oh, sorry. So these five were the integration of artificial intelligence and machine learning, because again, as we said before, with integration of artificial intelligence and machine learning, we might have the issue of potential biopiracy, taking up data from different sources that might not have a declaration of geographic location, that might not have consent from different communities to be used, and also might not be into a mechanism of potential benefit sharing and other uh, concerns that would arise. And all of them are included in the report that I'm going to share a little bit later. And also inequality in the participation of developing countries. As we know, right now, most of the sequences that are used and most of the developments that are produced are not in developing states, but in developed states. And this is an issue because some of these applications that developed states um, do are tested in developing states because of lack of understanding maybe of these mechanisms, lack of understanding and lack of knowledge transfer, capacity building, and technology transfer to assess these new products that are coming up. Also, not only this inequality on groups, for example, when women may not have equal participation like men, youth like other generations, IPLCs on regards of of other populations. So like you see here, we arise issues that are also cross-boarding issues. We have here also self-spreading vaccines for wildlife, which is very important for the fight of different diseases, but also might affect some populations that are not target. Self-limiting insect systems, and the development of the engineered gene drives to control vector-borne diseases and invasive species, which is another concern that we have because if we want to use these tools to control vector-borne diseases and invasive species, we might expect only to target one species, which is a target species, but actually no. As of right now, we are seeing concerns regarding off targets species, which might be important for example, pollination or preservation of traditional lands and practices and customs. So in this context, we need to start to discuss and make deeper assessment. And it's one of the things that is also going to be discussed at COP, how we can make deeper assessment into these 5 points to come with solutions and probable policies that can help parties to enhance their capacities into these points, basically, and issues. So here are some of the documents that you can read that came from the multidisciplinary expert group that were also used by SAPSTA for the recommendation that is gonna be in the table at COP16, basically. And this is gonna be the recommendation adopted, which is gonna be 26/4 in synthetic biology, is gonna target some of the issues that we spoke and the deeper assessment of these concerns, and also the continuity of the multidisciplinarity and also the multidisciplinary expert group as a whole. So this will be everything to understand a little bit in a thick mode the synthetic biology concerns that we can have right now. I made a little pause and I hope you did a little bit pause because I know and I understand these issues might be a little bit technical, more than other issues, but they're important because we need to understand the implications that this is gonna have for the youth. And we can have more discussions about the implications later on at the end of this presentation. And now we're gonna go to the next part of this, which is gonna be risk assessment, agenda item number 6. So, what is basically risk assessment? Or the context of risk assessment and management. So in the session CP10 of 10, the meeting of the parties that serves as the convention of the parties of the Cartagena Protocol agreed to have additional voluntary guidance to support case-by-case risk assessment of modified living organisms contain engineered gene drives in accordance to the Annex 3 of this protocol. Also, these materials should have a special focus on different organisms, especially living modified mosquitoes that contain engineered gene drives, as we see as we saw before. But not only this, also another one regarding fish. And this is going to be something that we're going to be discussing later on. So, as we saw, I'm not going to explain a little bit more, but just to refresh, that gene drives actually might have a higher percentage of transmission than normal alleles that go under Mendelian laws. So this is also something that has been considered in the risk assessment arena. And from this, we're gonna have to understand the different definitions between risk assessment, risk management, and risk characterization, which is basically what we do. So when we go into a field and we want to apply, for example, a new organism that has been, for example, genetically modified, either if it's a GMO or not, we need to make a risk characterization, which comprises risk assessment, meaning that is going to be more of the scientific and biological part, comprised with hazard identification, meaning identifying what is this organism, which is the modification that it has, what is the effect on this organism that is going to have this modification, how can we assess the exposure, and how can we assess the dose response of this organism into this environment that we're going to be inserting into. Also, in the risk assessment arena, we determine more technical stuff, such as, for example, the rate of reproduction, how it's going to be spread throughout the field, and more. In risk management, what we want to do is to how we're going to control this potential hazard that is going to be into the environment. Risk management, independently of risk assessment, is a little bit more um, political. It's a little bit more political. It's a bit more of policies and legislation that we have right now. And within risk management, what we want to do is to make control decisions, decisions on how to control this hazard. We are gonna determine acceptable levels of risk of this organism, and also we're gonna have some alternatives that can be placed into the field in order to control this organism. And this everything is going to be what comprises the risk characterization that parties need in order to determine whether or not an organism can pass or not into their controls and can go into their borders inside and be used, commercialized, consumed, etc. And this, of course, has a feedback that is not static, it's a process that goes and feedbacks by itself. And parties submit their risk assessment in different organisms into mechanisms that the CBD have implemented, like, for example, the clearinghouse mechanism. So, this risk assessment and risk management decision actually recalls the decision CP 913, where the ACTEC developed different guidance materials on different organisms, which are living modified organisms containing engineered gene drives and living modified fish. That was called from the parties to have these guidance materials because the decisions to take in order to have risk assessment of livified or modified organisms such as fish is highly important because fish have high mobility rates. They are not static in one place, and they can move from one jurisdiction to another because of how the governments of the sea and the governments of body waters, water bodies of water occurs. So in 2016, the Secretariat published like general guidance into the assessment of LMOs, which include mosquitoes, but it didn't specify guidance or didn't have guidance that is specific for engineered gene drives in LMOs, which was something that parties were starting to lack. Another thing that was also seen into this was the use of the precautionary approach, which is something that we as youth push because the precautionary approach is what is going to allow us to first analyze and understand which is that is going to be inserted in the field before is being inserted. And the use of the precautionary approach allow us to maybe be used in the, for example, lack of scientific certainty due to insufficient and relevant scientific information to understand the potential adverse effects of having these living modified organisms in the conservation and taking into account, for example, risk on human health. So, for example, if a party sees that there is not enough scientific certainty about genetic modified organisms or living modified organisms with engineered gene drives, it might take the decision through the precautionary approach to block the entry, export, the use, the commercialization of these modified organisms. And it's something that we need to push more. So risk assessment in SBSTA is defined by this two documents. One is the recommendation adopted through decision 26.5 that comes from, of course, the ad hoc technical expert group in risk assessment that had a meeting this year in 2024. So basically, all of these is managing different and additional guidance material that needs to be included for parties to assess these new, for example, technologies that are arising. And it's something that we need to push as youth, because through these, we can ensure that one, the precautionary approach is going to be used. Countries, stakeholders, organizations, and other groups, I can make informed decisions. We can push for example, capacity building, technology transfer, not only this, knowledge transfer to different stakeholders in order to understand how these new and potential organisms that are going to be released into the field, that are released into the field, might affect biodiversity in the short, mid, and long run. So this might be everything for today. I recommend you to have a little read into the documents to see how is the direction of the discussion into these questions. And if you have any questions, do not hesitate to contact the Given Policy team, and we will be more than glad to navigate together through these topics. So thank you so much.